Welcome back to the structure and function of bacterial cell. I welcome you back to the structure and function of bacterial cell. The last time we met, we spoke about bacterial strains and the four categories in which they fall into in terms of the shape. And we spoke about the spherical or cocci, and we drew it. Then we spoke about rod-shaped or bacilli. We spoke about the spiral, and we spoke about vibrio, the filamentous, and the common shapes. Usually, when we have spherical bacteria that occur singly, we suspect some organisms based on the gram stain and the oxygen relationship. If the organism of interest, the bacterium of interest, if the organism is gram positive and is aerobic and is a cocci, then we suspect staphylococcus if it occurs in clusters. If it is gram positive and the oxygen relationship is also aerobic and it occurs, the spherical shapes occur singly, we suspect micrococcus. If the bacterial cell that we observe under the microscope is gram positive and aerobic and it occurs in chains and it is a sphere or cocci shaped, then we suspect streptococcus species. This does not mean that all organisms that are spherical are going to fall into these categories of micrococcus, diplococcus, streptococcus, or staphylococcus. This is because the gram reaction may be positive or negative. If the gram reaction is negative, then it cannot be micrococcus, it cannot be streptococcus, it cannot be staphylococcus, because these organisms, this species, this genus that I just listed are all gram positive. So I'm going to give you an assignment before I continue, and I'm going to allow you to do the assignment in the next 1 hour, and you are going to submit the assignment back to my office or through your class rep, and then you can continue with the lecture. So after you've taken the assignment, you can take a break to do the assignment, and then you come back to continue with the lecture, the remaining part of the lecture. Now, the number one assignment is that you are going to list 5 genera of bacteria that occur, they are spherical or cocci, and they are gram positive and they're aerobic. That's the first one. Aerobic bacteria, gram positive, and cocci. Number 2, you are going to list 5 bacteria genera that are gram negative, aerobic, and cocci. The cocci can be any of the shapes. Once it's spherical, you list them in that group. Number 3 is gram positive bacteria that are anaerobic and they are cocci. Gram positive bacteria that are anaerobic and that are cocci. Number 4 is gram negative bacteria that are aerobic. Gram negative, anaerobic, and cocci. So you list 5 of such, and then we can come back immediately and continue with the lecture. So last time we met, we spoke about different things. We spoke about the bacterial colony and some things that we have to look at when we are characterizing bacteria. Today we are going to continue, and then from there we move on. I spoke to you about the bacterial colony, that it is discrete, pigmented. So when you see the bacterial colony on the petri dish plate, you look at it. Is it discrete? Is it pigmented or colored or chromogenic? And then that time I asked us about the skin color of the class rep of microbiology, 300 level. And some of us said it's dark complexion. Some say it's fair. Some say it's chocolate. But all these that I just said are not colors. If you look at the list of colors, those are not colors. The color can be black, it can be white, it can be yellow. I also called a lady that was sitting towards the door in the new SLT1. And I asked us what is her skin color. Some said she was fair, some said she's this, some said she said many things. Now I said that is not color. Those are not colors. Her color is yellow. So when we're talking about color, you have to know what you're saying. Then on the colony, the bacterium can be shining, it can be glistering, it can be opalescent. Then you look for the spreading of the colony. Does it grow more rapidly and cover the surface? If an organism grows more rapidly and cover the surface and you want to study it, you don't have to wait for 24 hours. You can wait for 4 hours. You can wait for 8 hours to see how far it has spread so that you can study the organism properly. Then we have the pinpoint colony that are very tiny and not easily seen. Then we also spoke about the surface of the colony, that it can be described as concave, convex, or flat. And it can be smooth and it can be rough. Then we moved on to the cell membrane, and I told us about the bacterial cell membrane. Now, under the colony, I told us that in characterizing the colony, you do not really have to depend on your memory. There are standard texts that you can use in describing the colony and characterizing the bacterial colony, both on the plate and under the microscope. The tests that you're going to do, the various tests that you're going to do also, they can be done with the aid of standard texts. The standard text, you just follow the procedure and you observe the results. Today we are going to continue, and what we want to look at now is the cell wall. We are going to look at the cell wall immediately now. The cell wall immediately surrounds the cytoplasmic membrane. Without it, all cells will be spherical. This implies that the cell wall determines the shape of the cell. It physically protects the cell membrane because the cell membrane is susceptible to physical and osmotic lysis. The chemical composition of the cell wall is important in distinguishing bacteria from other protists, as well as among groups of bacteria. Most bacteria produce one of two types of cell wall. That is, a bacterium can be gram-positive or it can be gram-negative. Structurally, the cell wall is complex. It is a rigid external layer whose strength is due to the possession of peptidoglycan. Peptidoglycan is also called murine. Peptidoglycan is spelled as Peptidoglycan, and it is also called murine, M-U-R-E-I-N, or mucopeptides, which is found only in prokaryotic cells. It is a large molecule consisting of amino acids and sugars. The sugars are N-acetylglucosamine, NAG, capital NAG in bracket, N-acetylglucosamine. Acetyl is ACETYL, GLUCOSAMINE. So I said the sugars are N-acetylglucosamine and N-acetylmuramic acid. N-acetylmuramic acid. Muramic acid is M-U-R-A-M-I-C. And N-acetylmuramic acid in bracket NAM. These sugars alternate to form a long chain attached to a N-acetylmuramic acid are side chains of 4 to 5 amino acids, making a cross-linked structure that covers the entire cell. The amino acids are L-alanine, L capital dash alanine, A-L-A-N-I-N-E. Then another of the amino acids are D-alanine, capital D, then alanine, capital D dash A-L-A-N-I-N-E. Another of the amino acids are D-glutamic acid and lysine or diamino phenolic acid, DAP. Some bacteria have several layers of peptidoglycan. The inability to produce this substance by a bacterium may result in a defective cell wall. So any bacterium that does not produce a cell wall may be a defective bacterium. In fighting bacteria, some antibiotics like penicillin act by preventing the synthesis of peptidoglycan and thereby induce cell lysis. You know, when peptidoglycan is not produced, the cell wall will not be able to be produced, and then the cell is going to disintegrate. Now, let's talk about the gram-positive cell wall. The gram-positive cell wall is stronger because it has thicker and several layers of peptidoglycan. In addition, it contains teichoic acid. Teichoic acid is TEICHOIC. Another auxiliary molecule that influences the ability to cause disease is the cell wall associated M protein that is found in some bacterial cell wall, e.g. Streptococcus pyogenes. This protein and the teichoic acid consist major surface antigen. So this is what you have here. The cell wall can determine if an organism is going to be pathogenic or non-pathogenic. The gram-negative cell wall is more fragile because it has a thinner peptidoglycan layer. The outer surface of the peptidoglycan is covered by a protective coating made up of lipoprotein and a lipopolysaccharide. The staining behavior of these two types of cell wall are different, and the differences are due to the variance in the physical structures of the cell wall and their chemical composition. There are some bacteria that are called wall-deficient variants. They are called wall-deficient variants. Only one group of bacteria, that is Mycoplasmas. Mycoplasmas is MYCOPLASMA. Mycoplasma naturally exists without cell walls. However, wall-deficient or deficient forms are known to develop among other types of bacteria. These wall-less forms are called L forms. If a cell loses its cell wall, the resulting structure is called a protoplast. A bacterium without a cell wall is called protoplast. Those with a weakened but intact residual cell walls are called spheroplasts. Now, let's leave the cell wall alone and let's move to the capsule. You can check out the difference between the gram-positive cell wall and the gram-negative cell wall and tabulate it in your notes at your spare time. Now, let's move on to the capsule. Most bacteria synthesize organic polymers, that is, exopolymer, which are deposited outside the cell wall as a loose amorphous layer attached to the cell wall, which is referred to as a slime layer, dense coat, or capsule. A different structure also exists in some others, called glycocalyx, GLYCOCALYX, which is a tangled mass of thin polysaccharide fibers that extend from the bacterial surface. These structures are not essential to the cellular function, and hence many bacteria do not produce them. Even those that do can lose this ability due to nutrition without any effect on their growth. All capsules are made up of polysaccharides, and they can exist in form of cellulose, as found in Acetobacter, or they can also exist as cholic acid. They can also exist as cholic acid, as found among enteric bacteria, or as dextrans among leuconostoc, or as leaves in Pseudomonas. Now we're going to move on to the next explanation. The amount of capsule produced depends on the growth environment. For example, polysaccharide capsule producing organism will produce more in media containing sugar. Just like microorganisms in the body, we produce more capsule material than when outside. The ability to produce capsule is also a genetic trait that can be lost, however, by mutation. Capsule-producing bacterial colonies are called smooth strains because of their surface texture. Uncapsulated ones have dry, irregular surface and hence referred to as rough strains. The capsules may play major roles in the cells that possess them. One of these roles is protection against desiccation from drying. Secondly, it contributes to the cell pathogenicity, that is, the ability to cause diseases, in that they protect whole cells from phagocytosis and destruction by white blood cell. In doing this, the capsule or glycocalyx cement cells together to form aggregates that are too large for the white blood cells to engulf. The glycocalyx also make adherence possible, which is an important factor in the development of some diseases, particularly urinary tract and gastrointestinal infections. The development of dental caries depends on the attachment of Streptococcus mutans to the tooth. Bacterial slime or capsule forming is a major nuisance in industries, particularly dairy, and paper industries. So this is what we have to mention about the capsule, and we need to still check. You need to check other materials to get the full details of the capsule. The diagram of the capsule is being displayed as I explained. You have to check it out and draw it also. Now I want to talk about the bacterial genome. The bacterial genome is the next thing we want to talk about now. The genetic information in bacteria is carried in the nucleoplasm in a structure called nucleode or bacterial chromosome. Nucleode is double-stranded helical DNA molecule unassociated with basic protein and is circular and attached to the cytoplasmic membrane. This single chromosome carries all the genetic information necessary to specify the essential properties of the cell. Many bacteria can also harbor small extrachromosomal circular DNA molecules called plasmids. The plasmid is capable of autonomous replication. That means it can divide without reference to the cell, and they carry determinants for such phenotypic character as drug resistance and enzymes, which mediate certain peripheral metabolic pathways. The amount of DNA in the plasmid is usually 0.5 to 1% of that in the bacterial chromosome. In E. coli, cells undergoing rapid growth have more chromosomes per cell than those undergoing slow growth. During rapid growth, nuclear division typically runs ahead of cell division. Segregation of the genome is achieved by a mechanism which does not involve the complex sequence of structural events associated with mitosis. Segregation of daughter chromosome is brought about by the growth of the membrane between two attachment sites. During cell replication, therefore, each daughter cell contains two already separated chromosomes. Bacteria normally exist and reproduced by asexual means in the haploid state. A diploid state can arise transiently in prokaryotic organisms as a result of genetic transfer by special mechanisms during which there is a unidirectional passage of DNA from a donor cell to a recipient cell. These special mechanisms include the following: conjugation, transduction, and transformation. When you come to microbial genetics and the methods in microbial genetics, we are going to deal more detail with transduction, conjugation, and transformation, and also genetic recombination will be spoken about, and the more details will be taken at that point. But at this point, we need to know that conjugation takes place when there is a direct cell-to-cell contact. The cells must be in contact for conjugation to take place. Transduction on its part is mediated by bacterial viruses called bacteriophages, which incorporates fragments of the genome donor cell. Transformation is mediated by free DNA fragments derived from the donor cell, but which passes through a medium but taken up by the bacterial cell. In transduction and transformation, only small fragments of the donor genome are transferred, while the entire chromosome genome can be transferred in conjugation. So you can see the difference in transduction and transformation. Only small fragments are transferred, but in conjugation, the entire chromosome genome can be transferred. You have to know that there are small parts or small fragments that are transferred in transduction and transformation are enough to cause genetic change in the organism. So, whether it is small fragment or large fragments, they need not be taken for granted. And in fact, it is the small fragments that lead to mutation and other things, changes in the genetic information that makes them to vary from cell to cell. Bacterial appendages are the next thing I'm going to talk about now. They are the flagellum. Those are the bacterial appendages, flagellum. Appendages are something that is attached to something. The main thing is there, appendage is attached to it. So bacterial appendages are attachments. One of such attachments is flagellum, another one is the axial filaments, and another is the pillar. The location and number of flagella on a cell and the number of waves of an individual flagellum may vary according to species. Various types of flagellation are recognized. Number one type of flagellation is monotrichous. Monotrichous is spelled monotrichous. Monotrichous flagellation means the organism, the bacterium, has one flagella. So when you draw an organism like a bacterium, you can draw a sphere or a cuboid to represent a bacterium, and then you draw out just one flagella from that bacterium. That is monotrichous. Mono means one. So we also have lophotrichous. Lophotrichous means it has more than one, but it is in one direction. So at the point where monotrichous have just one flagellum projecting and moving out in one direction, lophotrichous have three flagellum on the same side. That is lophotrichous. And then we also have the amphitrichous. Amphitrichous means amphi from the word amphi as an amphibian. Amphibians can live in water and they can live on land, so they can live on both water and both land. So the same vein, amphitrichous means the flagellum, amphitrichous flagellation means the flagella is present on this side and on that side, on both sides, as you have in amphibian. They can live on both water and land. So in this case, the flagella is present on this side and on the other side. So if it's present on the right, it will be on the left. If it is up, it will also be down. If it is on the anterior, it will be in the posterior. If it is on the lateral side, it will be on the first part of the lateral side to the right, and then it will be on the left. That is flagellum. These three flagellum, the arrangement of the flagella I've mentioned, are called polar flagellation. Polar flagellation because they are found at the ends. They are at the ends, and they can help its solubility in water. They can help it navigate. So they are at one end, polar. This side, that side, polar. Then the fourth type of flagellation is called peritrichous. peritracheous. Peritracheous is spelled P-E-R-I, tracheous. And this occurs all over the body. So if we draw a shape of a bacterium, in any way we draw the shape of the bacterium, then the flagella are many, and they come out from this side, that side, that side, that side. On this side there's flagellum, on the upper there is, below there is, on the right side, on the left side, and in between there is flagella. That is peritracheous flagellation. The diagram and the labeling of the flagella is projected now. So I want to take a closer look and to also go back and practice the diagram and the labeling of the diagrams. Flagella are rigid and unlike the flexible eukaryotic counterparts, do not weave forth and back. They are rigid. Instead, they spin much like a propeller of a boat. The direction of the movement is usually determined by chemotaxic. It is made up of three parts, essentially: the filament, the hook, and the basal bodies. Flagella are rarely observed among cocci. They are rarely, not that they do not exist, but they are rare among cocci. So you can check that out on your own. Exafilaments are found among the spirochetes. Spirochetes is a group of bacterium that have specific characteristics. Spirochetes is spelled S-P-I-R-O-C-H-A-E-T-E-S. So exafilaments are found among the spirochetes. We mentioned initially that we have bacterial appendages, flagellum, exafilaments, and pili. So we are looking at exafilaments now. They are found among the pyrochetes. It is composed of two fibers that originates points of the cell to overlap in the middle. They originate from parts of the cell to overlap in the middle. Unlike flagella, exafilaments are covered by the cell wall. The organism moves by rotating along its longitudinal axis and by flexing and bending along its length. Another of the appendages the bacteria has is the pili. Pili are hollow tubes that extend from the cell. They play no role in motility. They are straighter, shorter, and thinner. They are found only on certain species of gram-negative bacteria, usually in large number. So you can check online if you have any gram-positive bacteria that have pili, because it has been stated here that they are found only in gram-negative bacteria, and they are usually in large number. So these are some of the bacterial appendages and how they do. So diagram of the flagella and exapili and flagella, exafilaments, and pili are shown on the board. So I've mentioned that the pili are straighter, shorter, and thinner, and that they are found on certain species of gram-negative bacteria, and they are usually found in large number, not in small number. So I want to look at the function of the pili now. What are the functions of the pili? What are the functions of the pili? Because they have their own function. Pili helps in conjugation, that's the first thing, between bacteria, and they also help in subsequent transfer of information, that is copies of chromosomes or plasmids. This process requires special pillars called F, or fertility pillars. The F is capital F. So during exchange of genetic material, pili is important. Pili are also used for attachment to surfaces, such as infected persons. Therefore, pili, like the capsule and glycocalyx, contribute to the establishment of infection by binding bacteria to cell surfaces in areas where they would otherwise be eliminated by the movement of body tissue. So these appendages help bacteria in its pathogenicity. They help bacteria to attach to surfaces, and when they attach to surfaces, they cause infections. We'll move on now to look at endospores. What is an endospore, or what are endospores? Endospores, a number of bacteria can change into dormant structures that do not grow or reproduce. These structures are called what? Endospores. They are dormant. And most of the time, endospores are formed when conditions for growth and reproduction are unfavorable. And endospores are resistant to heat. They are also resistant to radiation. They are resistant to chemicals, and they are resistant to various agents that may be harmful to vegetative cells of bacteria. Therefore, endospores protect the cell from adverse conditions. However, the endospore, unlike their counterparts among eukaryotic organisms, is not a means of reproduction, since there is usually no increase in the cell number during the spore cycle, meaning that a single bacterium is formed from a single spore. A single bacterium is formed from a single spore. So endospore is not a means of reproduction. It is just a means to escape unfavorable condition. When the condition becomes favorable, a single spore will form a single bacterium. From the spore, an actively growing vegetative state can be restored by germination. Sporulation is a response initiated by depletion of an essential nutrient in the environment. During the formation, the cell membrane encloses a section of the cytoplasm containing the entire chromosomes, ribosomes, and other soluble cytoplasmic materials. This section is called the core, C-O-R-E. Subsequently, a series of relatively impermeable layers develop around the core to protect it from damage. The diagram of these are going to be made available to you as we go on. In fact, you can see the diagram on the diagram being shown. During sporulation, a cell loses most of its water, which eventually accounts for its resistance to heat. But upon germination, the cell takes up water and enlarges. The spore coat disintegrates and vegetative cell emerges. The position and shape of the spore within the parent is characteristic of the species and the age or health. Microscopic identification of bacteria. There are three forms and shapes among the bacteria that possess spore. Number one is the location of the spore itself and the bacteria. The location can be central, where the spore is located in the center of the bacterium. So it can be central. The spore can be said to be central. The shape is shown. It's shown now and projected. Then the spore can be terminal. Terminal. A spore is like a protrusion. So the spore can be terminal where it occurs at one end. Then the spore can be subterminal where it is not in the center and it's not at the end. It is subterminal. It is not at the end exactly and it's not at the center. So it's between the center and the end. That is subterminal. Spores are formed by members of the genera Bacillus. And Bacillus are aerobic. They form aerobic. They are aerobic. So aerobic spores are formed by members of the genera Bacillus, while members of the genera Clostridium form anaerobic spores. So you can find out other genera that produce spore if there is. So we are going to end this lecture at this point, and then we're going to continue in our next class. You need to check the diagrams and see and practice the diagrams of the spores that we mentioned and the location and the formation of the spore and every other thing that we need to know about the spore. So you need to also take notice of every aspect that I've been spoken about. In the next class, we're going to be looking at something else, which is bacterial growth. In the next class, we're going to be looking at bacterial growth and bacterial growth requirements. I'm going to talk to you some other time. If you have any question, you can ask your question now, and then we can take the question together. I need to state here that you need to check other materials, check the textbook. We have different textbooks in microbiology. One of them is Prescott Microbiology. Another is Nesta. Another is Brooks. Another is Talaro and Talaro. And many other texts that you can check both online and offline. So try and check all this, and then you are going to be glad you did. So you have to note that the difference between gram-positive and gram-negative cell. Also, we know that Mycoplasma does not have cell wall, and there are other bacterial cells that are defective, and the cell wall is gone. So we're going to continue in the next class, and if there's anything that is not clear, we're going to make it clear to you more in the next class when you bring up the question. So I want you to go over the materials I've looked at, and then you try to form your notes more based on these materials, and then try to practice giving yourself some questions and answering them.